Hello everyone, let us continue with our lesson. We have already learned different attacks for aldehyde and ketone. We have 2,4-DMP for both aldehyde and ketone. We have Tonens reagent, valence reagent for aldehyde only, and then we have an autoform test for testing the specific two groups. Okay, I hope you can remember all of them. And it comes to the last part of our syllabus, which is very easy. It's called infrared spectroscopy. So same as before we go back to the lecture notes, I give you an introduction on what it is, okay? Basically, it's used for finding the functional groups, identifying the functional groups of an organic compound. So the working principle is that we use a part of the EM wave to radiate into the organic molecules, and this part of the EM wave is infrared. Okay, infrared. If you study physics, you know what it is. So because the functional groups of organic molecules are very sensitive to infrared spectrum, different functional groups will absorb a particular wavelength, okay, a particular wavelength from the IR to undergo vibration. So by detecting the remaining IR after passing through the organic molecules and see which parts are being absorbed, then we will know which functional groups they have. Do you understand what I'm saying? So for example, if you have a organic molecule, okay, and then you use IR in, and then after that the IR will come out. Then by looking at which part is absorbed, you know what kind of functional group it has. For example, if it absorbs around this frequency, okay, then we know that it contains a carbonyl group. If it absorbs around this frequency, then we know that it contains OH. Basically, these two are the most common frequency that the exam paper will ask you. Okay, then what's the use of the IR spectrum for the functional groups? Why do they absorb the IR? Because for each bond, for example, this bond, as I said, it absorbed the frequency around this one. Now you don't have to remember this data because it is given in the data booklet, okay? So different bonds will absorb a particular frequency for vibration, okay? Because this is we call it the lateral frequency for this bond, okay? Or you can call it resonance frequency. Resonance. How to spell? Resonance frequency, okay? After absorbing this part of the frequency, this bond will undergo vibration in three ways, okay? The first way it can be done by bending. For example, if you have a bond like that, bending means it's going in and out, bend itself, and then it can also absorb the frequency by stretching. Stretching means the bond become longer and shorter, and then the last way of vibration. There are three kinds of vibration. Is twisting. Twisting is they twist together. For example, this one goes to there, this one goes to here. But anyway, by doing this, this particular frequency from the IR spectrum will be absorbed, okay? Then by looking at the IR spectrum, you will know what kind of functional groups does this organic molecule have. I will go through some IR spectrum with you later, okay? Like this is the introduction. Okay, now then let's just go back to the lecture notes. Let's just go back to the lecture notes. And fill in some blanks together first before we look at the IR spectrum together. Now, can you see this will be given in the exam. This will be given in the exam. This one in your data booklet. Okay, you don't have to remember anything. Normally, this is very important and this is very important. Now, not only you have to pay attention on the number. Now, the unit is cm-1. Not only you have to pay attention on the number, you have to also pay attention on the shape. For example, this one is counter sharp. I mean the peak, the absorption peak. And this one is counter broad. Okay? Now we can go and look at the IR spectrum together. For example, these two. Now let's take a look at the question. The last page. Explain if the IR spectrum of A or B correspond to the one produced by ethanoic acid. Now when you take a look on ethanoic acid, right? So now it contains CO bond and OH bond. As I mentioned, when you go back to here, the CO bond will give a sharp peak around this number. And the OH bond will give a broad peak around this number, right? So let's take a look. Now, for this one, it has a very broad peak. Can you see that? We call it absorption peak, okay? It goes down and then you can see the trough because it's being absorbed at this part. Okay, absorption peak, broad absorption peak around 3000. Okay, so this can tell you that it contains an OH group. Understand? And when you look at here, around 1700, there is a sharp absorption peak. So it also tells you that it has this group, right? So since since it both contains C double bond O and OH, so obviously this is the answer. Now you can look at the later one. The later one, even though it has an OH peak, okay, the broad absorption peak around 3000, but when you take a look here, okay, there is an absence of the sharp absorption peak. So which means that it doesn't contain C double bond O for this one. So the answer is A, okay? Then when you try to put the answers together, you can say, of course, the answer is A lah, because it contains a broad absorption peak at around 3000 cm-1, which corresponds to OH group, okay? And it has a sharp absorption peak at around 1700, so which corresponds to C double bond O. You can also mention for B, there is absence of a sharp absorption peak at around 1700, right? So there is no C double bond O. Therefore, the answer for ethanoic acid will be A, because ethanoic acid contains both the C double bond O and the OH. Do you understand what I'm saying? Oh, I still have some time. Let's fill in the blanks together. I will give you more exercise later, don't worry, okay, for this chapter. Oh, exercise. Sample being analyzed is irradiated with electromagnetic waves in the infrared region of the electromagnetic wave. Now the machine used is called spectrophotometer. Okay, can you please remember the name of the machine? Of course, we don't have this kind of machine in the chemistry lab. It's a very advanced machine which can be found in university. There is a typo, detects, okay, detects the intensity of the wavelength of infrared radiation that passes through the sample. Now, resonance frequency, I mentioned that, is the particular frequency of the infrared absorbed by molecule for vibrating by stretching, bending, and twisting. Okay? So different functional groups at different resonance frequency absorb. Okay? Do you understand what I'm saying? Okay, now let me give you a summary. Today we are talking about the IR spectroscopy, that is to irradiate the organic molecule with the IR spectrum of the EM wave. Different functional groups will absorb a particular region, a particular part from the IR spectrum. By taking a look on which part is absorbed, you can deduce which functional groups they have. Most likely in the exam there are two peaks that always appear. One is the C double bond O. You will see a very sharp absorption peak around 1700. The other one is the very broad peak around the OH, and it is broad because the OH can interact with each other to form hydrogen bond. So it makes the shape of the peak bigger, right? Because more energy is required to overcome the hydrogen bond link with each other for vibration. Okay, and then you can see this one corresponds to ethanoic acid because it contains both the broad peak and a very sharp peak here that corresponds to the OH and correspond to the C double bond O respectively. But for B, you don't see a sharp peak around 1700. Okay, let me stop here, and then later on I will find more worksheets of IR spectrum and practice and discuss the questions with you.